So, you guys happen to know what time it is?
No, what time is it?
It is practice presentation time!
Yes! Yes! It's go time! So remember that lagers are ferocious, but they are F1 sterile hybrids, so they have absolutely no evolutionary implications. However, a lot of hybrids are not sterile, and therefore do go on to produce F2 and F3 in later hybrid generations. When that happens, you can get a phenomenon called transgressive traits, which means that the hybrids produce traits, quantitative traits that are either higher or lower than either of the parental phenotypes. You can see that demonstrated here, where the parental phenotypes only have 2 or 3 flowers, whereas the hybrid's going to have 1, 2, 3, or even 6 different flowers. And this can happen for any type of quantitative trait in hybrids. And this produces variation that can be acted upon by selection to produce hybrids that are well adapted to novel situations and novel environments. Another interesting thing about later generational hybrids is that they can produce gametes with quite a bit of variability, much more than their parents' gametes, the variability that you would find with parents' gametes, which means that they produce a lot of variation, again, that selection can act upon. And in connection with transgressive traits, this creates quite an environment for selection to act upon. So for my studies, I'll be doing 2 greenhouse experiments in which the first one deals with F1 hybrids. See if F1 hybrids have a greater host range than their parent strains. So I'll be using Mycobacterium on Silene and Dianthus for that experiment. My other experiment concerns later generation hybrids to see if they can, over multiple generations, evolve to infect new host species better than the parent non-hybrids can. And I'll be doing that by propagating different strains, non-hybrid and hybrid strains, on novel hosts over multiple generations and picking the best individuals from each generation and using those as the inoculum for the next generation, and that way see if they can see which type of pathogen strain can adapt best to a certain type of novel host. And that's about it. Thank you.
Alright. I'm going to be working on Silene.
Question? Question?
We'll be working on Silene.
Question.
Yes?
Which host genus is better, Dianthus or Silene?
Well, we've worked with Silene a bit longer, so we know it better. And the species of the pathogen on that host genus are more clearly defined.
Wrong. Dianthus is the answer. Dianthus is better because the plants have radiated more recently and therefore allow more pathogen hybridization.
Okay. Thank you, Dan.
In addition to my greenhouse experiments, I plan to investigate wild populations of Dianthus in order to look at the frequency of hybridization and sympatry of its pathogen in the wild, as well as to assess the level of genetic heterozygosity within and among different pathogen populations. This will allow me to look at the evolutionary consequences of different pathogen hybridization events in the wild. I think Aaron feels threatened by my audacity.
Alright, continuing. I'm working on Silene meridema, and you can basically break up my thesis into 2 components, disease dynamics and phylogeny and systematics. And so I'm basically taking a 2-pronged approach to investigating why the sympatry occurs. And so under disease dynamics, we're going to go sampling populations and see if the disease-free populations are resistant or susceptible, so where they are in the disease cycle and whether they've already overcome the disease or the disease has yet to arrive in that population. And then we're going to see if there's actually a cost of resistance upon the fitness of the individuals. And we can do that through greenhouse experiments and seeing if there's lower seed production or stunted growth. And we're going to see at the individual level if one species is resistant to one species of the host is resistant to one species of Mycobacterium. Is it also resistant to the other one? And we're going to also look at the differences in the anthers of the 2 different Mycobacterium species and see if that comes from genetic variation of the host or of the pathogen. And so we can basically clone the same genotypes of the host and see if you end up with different anther morphologies when you inoculate those. And so here You see the darker anthers are that of Inflate, and the lighter ones, as pale as down here, are that of Lagerheimia. And so with phylogeny and systematics, we're just taking a look at how the two species came to form this sympatric association, which might help us to figure out, I guess, if they're really in symmetry. And also we're going to see if the Inflate species of Microbotryum is different on Rhodoma than it is on vulgaris, which is another host plant. That's basically it.
Nice job, Aaron.
Thanks, Dan.
Which project is better? I don't know. I mean, they both have their merits. Dan's working on his hybrid thing, which sounds pretty good, and Aaron's got that sympathy thing. You know, I actually have no idea what we do in this lab. I've basically survived the past year on smiling and nodding, but check it out. Do I look like Bono?
Page 237. Side project number 4, aka Dan's thesis. It's already been done. By me. Same for Aaron's. Chapter 8, Part 5.
So I had a long talk with Michael, and guess what? I'm going to England! So yeah, basically we're just going to be sampling populations of Rhodoma along the coast, and we're actually going back to a site he went to last year. Here is Shingle Street. So I will be here and here at some point. So yeah, I'm pretty excited. Watch out, Dan. I'm going to be back with some awesome data.
She's going where? England? Michael's not taking me anywhere.